This is Dr. Peter Larson of the Loyola Genomics Facility, and I want to talk to you about some of the experimental design challenges that arise from next-generation sequencing. First, let's think a little bit about the humble p-value. Generally, while considering the results of an experiment with replication, a p-value less than 0.05 is considered significant. This can be interpreted as the probability of observing the result of an experiment as a consequence of random chance is 5% or about 1 in 20. That doesn't sound too bad, but what happens when the number of observations is more than 20? A lot more. For example, if the next generation sequencing experiment simultaneously measures the expression levels of 30,000 genes at once, that's the approximate number of genes in the mouse genome, what utility does a p-value of 0.05 provide? Well, for 30,000 measurements, considering a p-value of 0.05, you might expect about 1,500 seemingly significant observations that are due only to random chance. That's a lot of false positives. So, what do we do when the number of observations is very, very large? Well, you might start by increasing the threshold you want to consider significant. While this could cut down on your false positive results, it will come at the cost of increased false negatives, those relevant genes that you'll miss when you use a very stringent p-value threshold. Now, computationally, the solution to the problem of many observations is false discovery rate correction. FDR correction is a statistical approach that takes into account the number of observations and the observed distribution of experiment's p-values to find some balance between false positive and false negatives. While false discovery rate correction is an essential component to analysis of next-generation sequencing data, it's not by itself enough. Statistical significance is not the same as biological relevance. Just because you can identify the statistically significant differences between two conditions, it doesn't mean that comparing these two will lead to some greater insight to your experimental system. So how do you design experiments that attribute biological meaning to a list of thousands of genes? And the answer is good experimental design. So let's imagine the simplest of genomic scale experiments, comparing treatment A to treatment B. Let's assume that you performed all the relevant positive and negative controls and that your experiment identifies about 4,000 significantly differentially expressed genes. Is that any good? Well, a good first step is to make sure that your conditions result in measurable, quantifiable difference in phenotypes, so that here you can associate changes in gene expression with some observed biological consequence. All right, how about this phenotype? While there's definitely a phenotype to the treatment B, it may not be a phenotype that's specific to treatment B. In a gene expression study, you may detect events like stress response, damage repair, or metabolic remodeling that, while true, are a secondary effect of your experimental treatment and not a response to the treatment itself. The best response here may be to modulate the experimental dose or the timing of sample collection to capture the biological changes that are specific to your treatment of interest and not some later secondary or more general response to something like stress or damage. Okay, even assuming that you have the optimal dose and sampling time, how can you tell the difference between a response to an experimental treatment and some other non-specific effects? One good approach here is a matrixed or multifactorial experimental design. Here, we consider multiple experimental factors in combination and measure multiple phenotypic observations. Now we can more meaningfully associate gene changes with specific observed biological consequences, as in analysis of multiple variants, for example. Now, a final consideration for experimental design is biological replication. Because let's face it, you don't want to do any more expensive experimental replications you need, but if you don't do enough, you run the risk of performing an underpowered experiment that cannot distinguish between experimental effect and natural biological variation. If you know your desired statistical resolution and have some information on expected variance, you can use that information to identify how many replicates you will need for a desired statistical resolution. Now, while every experiment is unique and will bring with it its own experimental design challenges, thinking through these design elements for next-generation sequencing will help you get the most value for your research. And the best time to consider the most appropriate way to analyze your next-generation experiment is before you start the experiment. The Loyola Genomics Facility is available for consultation on experimental design so you can get the most out of your next generation experiments.